Hi, I'm Soohyun Jeong, a PhD candidate in the Mechano Lab at the University of Memphis. I'm honored to talk to you about my research, molecular phylogenetics, and evolution of Dorcasomini longhorn beetle with a focus on adaptive radiation and endemism in Madagascar. Dorcasomini is a tribe of longhorn beetles in the family Cerambicidae and subfamily Cerambicini. Dorcasomini are mostly distributed in the southern hemisphere. Interestingly, this group lacks a clear synapomorphy, and Dorcasomini from different continents are morphologically distinctive. Because of this, each regional group was initially treated as separate taxon. The Afrotropical group and Southern Palearctic and Oriental groups were considered as separate subfamilies, and the Malagasy species were placed in Leptrini, a different longhorn beetle subfamily called flower longhorn beetles. This is because of convergent evolution in adult morphology and flower visiting behavior. The Australasian taxa were misidentified or left unclassified until recent molecular phylogenetic studies redefined this group. These studies have redefined Dorcasomini as a single tribe Dorcasomini. These analyses also revealed that several Australian taxa belong within this lineage and should be included in this tribe. Another remarkable aspect of Dorcasomini is their distribution pattern. About 70% of all known Dorcasomini species occur in Madagascar, and every one of these Malagasy species is endemic to this island. And Dorcasomini make up only about 1% of global longhorn beetle diversity. However, in Madagascar, about 25% of all longhorn beetle species belong to Dorcasomini. This seems unusual, but this kind of extreme taxonomic bias is actually very common in Madagascar. For example, lemurs, the iconic dancing primates from movie Madagascar, and they are a well-known example of extraordinary endemic radiation in Madagascar mammal fauna. Dorcasomini appear to represent a similar pattern in the insect fauna, and this charismatic group arguably deserves iconic status comparable to lemurs. However, it remains surprisingly understudied. Madagascar is very important biodiversity hotspot. Its surface area makes up less than 0.5% of Earth land area. However, it harbors around 5% of species on Earth. And we can see extraordinarily high rate of endemism in this island as well. And here we can see the exceptional morphological diversity of Dorcasomini in Madagascar. They occupy a wide range of ecological niches in this island. As you can see here, there are some diurnal ones, mostly on the left side, and nocturnal ones on the right side. And most species are associated with flowers as adult, and their larvae feed on wood or rotten wood. And they usually pupate underground, but few are known to pupate in the tree. I highlighted some distinctive species appear to be similar to other insects. Hymenoptera resembling groups have shortened elytra and have shortened antenna even, and also they have very bright colorations. Other resemble different kind of beetles, like leaf beetles and even dung beetles. And even some species look like true bugs. So my research focuses on when and how this interesting group diversified in Madagascar. Specifically, where did the tribe originate? Where are the nearest living relatives of Malagasy Dorcasomini, and do they form a clade? And does the phylogenetic pattern of diversification suggest Dorcasomini adaptively radiated in Madagascar? I hypothesize that Dorcasomini originated in Gondwana. This idea is supported by the fact that most members of tribe are distributed in the southern hemisphere, which is the region that once was part of Gondwana. My second hypothesis is that Malagasy Dorcasomini went through adaptive radiation, showing an early burst pattern of diversification. The early burst model is one of the most well-established adaptive radiation models, and this is an example of adaptive radiation in anolis lizard in smaller islands. The graph shows accumulation of species through time, and we can see an early rapid diversification rate that later slowed down as available ecological niches become filled. I expect to see such pattern in Malagasy Dorcasomini, and to test these two hypotheses, I'm going to use phylogenetic analysis. One of the most challenging but also the most exciting part of studying insect group that is endemic to Madagascar is the amount of travel required to obtain research material. I traveled to major museums in Paris and Washington, D.C. to examine and borrow specimens. However, I encountered significant obstacles. Many older museum specimens yield low quality or highly degraded DNA. It made them difficult to use for molecular analysis. Fortunately, the Coleopterist Society supported a 2-month field expedition to Madagascar, which allowed me to collect fresh and high-quality samples directly from field and from institutions such as Madagascar Biodiversity Center and Center Bio. This trip also gave me a good opportunity to explore Madagascar's diverse ecoregions and also to build a strong networks both within the country and with the researchers worldwide who study Malagasy biodiversity. I aim to include Dorcasomini from all known geographic regions in my analysis, with particular focus on capturing the full diversity of Malagasy fauna. Most of the specimens have been obtained, and currently I am reaching out to personal collections to fill the remaining gaps in my dataset. For this talk, I built a preliminary tree using part of the data. These are mostly old museum samples. The method mainly focuses on effectively handling fragmented DNA from old museum samples. I used low coverage whole genome sequencing and used new pipeline Sapphire. The new pipeline Sapphire directly mapped raw sequencing reads to pre-made reference gene sets, which allows it to recover more loci and then make it comparable across different data sets, such as anchored hybrid enrichment data and low coverage sequencing data, and then genomic data. And this is preliminary tree based on 32 Dorcasomini species. The tribe Dorcasomini seems to share a common ancestor, and nucleotide tree shows monophylity of Dorcasomini. Malagasy group forms a single clade, and other groups from different continents that I sampled more than one specimens form a clade. Southern Palearctic and Oriental groups are more closely related to Malagasy group, while Australian and Afrotropical groups form another different clades. And this tree is not time-calibrated tree, so we do not know the exact timing of the branching event and hard to be conclusive, but the result aligns with my hypothesis of Gondwana origin of Dorcasomini. The Malagasy Dorcasomini are monophyletic, which is very interesting because it means the large diversity in Madagascar, as I mentioned earlier, 70% of Dorcasomini came from one common ancestor. And the shorter branch length in the Malagasy clade suggests that possibility of adaptive radiation. And when we focus on Malagasy taxa, one clade stands out. Even with limited species level resolution, this clade includes all members of Morphology Group 2 from Villiers et al. 2011, forming a distinct deep branch. And these beetles share derived characters such as shortened elytra and bright color pattern, and most of them are flower visitors. The preliminary data only includes low-quality museum DNA from common species, but I'm going to add fresh samples that I collected from field and other collections in the tree. This will improve species-level phylogenetic resolution and incorporate more representative samples of morphological diversity to detect evolutionary patterns. And then I will generate time-calibrated tree to understand biogeography and adaptive radiation. To sum up, each biogeographic group of Dorcasomini is grouped together, and Palearctic and Oriental groups were more closely related to Malagasy taxa than Australasian and Afrotropical groups. And lastly, Malagasy Dorcasomini are monophyletic and may have adaptively radiated in Madagascar. I appreciate all the support from our lab members and collaboration that made this research possible, and especially the Collaborative Society. The student research grant enabled me to carry out this work. And thank you so much for listening to my talk.